Good morning, everyone. It's really a pleasure to join you again in the fifth Minimal Cell workshop. It seems like yesterday, the first one we had. So my presentation today is entitled Engineering Minimal Phytoplasma Cells for Next Generation Crop Improvement. This is not exactly a project with results yet, but it's just a newborn project. And what I will share with you today is the rationale behind this idea. So, well, climate change, I think, is undeniable. We are already living its consequences. And one of the major contributors is the agriculture, especially through nitrogen fertilizers. So producing these fertilizers accounts for about 1 to 2% of global CO2 emissions. And their use releases nitrous oxide, which is a potent greenhouse gas and also a driver of ozone layer destruction. So a promising alternative is to engineering phytosynthetic organisms with nitrogen fixation genes. So, but unfortunately, this remains really, really challenging because nitrogen is extremely oxygen sensitive. So that is why we are considering hosts or engineering a specific host that can thrive in low oxygen environments and deliver fixed nitrogen directly to plants. So, but why are we doing this? Really, like, I think it's at some point also very challenging, but we are actually doing this because we have huge advances with the design of synthetic cells and synthetic genomes, mostly taking advantage of JCVI SYN3B. So it brings us a solid foundation to design new synthetic cells. Well, of course, and nature itself gives us some inspiration. So recently we have the description of the nitroplast, a cyanobacteria endosymbiontic that has evolved into a nitrogen-fixing organelle in algae. And of course, we also have mitochondria and chloroplast as well. And of course, our idea is to apply this logic to design a minimized bacterium that can live as a plant endosymbiontic and fix nitrogen or even other traits that might be interesting and serve as a natural biofertilizer. And one possible chassis for this is the phytoplasma species. So, but why phytoplasmas? Well, phytoplasmas have a very particular lifestyle. They are transmitted between plants by phloem feed insects and can survive inside insect tissues. So in plants, they live in sieve elements, which are non-nucleated cells that allow them to move through the phloem, and they secrete effectors proteins that reprogram plant growth and defense, preparing the host for long-term colonization, and even attracting insect vectors. So this complex cycle shows both the challenges and opportunities that we have in working with phytoplasmas as a synthetic biology chassis. So, but what is even more interesting about phytoplasmas is actually it belongs to the class molecule. So it's a cousin of the minimal cell. And their genomes are also among one of the smallest bacteria. So typically range from 507 to 1,000 kilobases, encoding only 500 to 1,000 genes. They lack many biosynthetic pathways. And if you take, for example, the Flavescent doré phytoplasma genome is only 654 kb with only 506 genes. So, and it shows low plasticity. So these characteristics makes phytoplasma interesting candidates for minimal genome engineering. So our conceptual model is what we call the phytosyn, which would be our phytoplasma synthetic or minimal phytoplasma. It will be designed as a safe, non-pathogenic minimal version of a phytoplasma, reimagined as a synthetic symbiote. So instead of producing virulence factors, so phytosyn would provide beneficial traits to plants, acting almost like a new organelle. And since this effort is being developed with Brazilian partners, so we affectionately gave it the nickname we are calling here in Brazil as feijoada, after a traditional Brazilian dish to reflect its origin. So for us, this is going to be our little feijoada. So, but how are we going to do this? How are we going to build this thing? So to design a synthetic phytoplasma genome, we are planning to combine comparative genomics and DNA synthesis. And the idea is actually to rationally minimize a selected genome, removing pathogenic effectors, unstable regions such as PMUs and transposomes, and keeping only the essential functions. So we can leverage DNA synthesis and yeast-based assemble for transplantations, for instance, once we have all this genome assembled. And we also plan to actually include a landing pad to add new genes in the future. But however, one of our major challenges is that phytoplasmas are difficult to culture in vitro. So we may use a hybrid system, and combine parts of phytoplasma genome with JCVI-SYN3B genome in order to overcome this problem if it actually comes to be an issue for us. And secondly, we are also exploring the use of AI to guide genome annotation. So comparative genomics between phytoplasmas and other molecules species can help us also identify the core gene set. So AI tools such as AlphaFold can predict the structure of hypothetical proteins and help us to clarify poorly characterized functions. And of course, together with metabolic models, this will allow us to make well-informed calls on genome structure and function, deciding which genes we have to keep or delete. So this approach will help us to design a genome that is minimal, functional, and of course, compliant with biosafety standards. It has to be no pathogenic at all for plants. Actually, you want to build something that actually helps plants and it's functional inside the plants. And we just started to make some analysis and comparing those genomes. So the first analysis that we performed is just to identify the core genes. We use T-BLAST search to annotate metabolic, structure, and other sectional functions across phytoplasma genomes. And here in this table, sorry, just in Portuguese, but we have the results of this initial comparison between JCVI-SYN3A transcribed proteins with the ones from different phytoplasma species. And it shows that at least 38% of the minimal cell proteins content is similar to phytoplasmas. So of course, this approach is showing that will help us to get our genome assembled. And once we have our genome built, we will pass to the next step, which would be to boot up our phytosyn. So booting up the phytosyn can follow three possible strategies. First, which is the one that I already mentioned, that is use JCVI-SYN3B genome as a receptor for phytoplasma minimal genomes or specific genes, mixing and introducing synthetic DNA with specific traits. Secondly, we're going to perform genome transplantation. So as Zuma just presented recently. And third, we can explore as well the bottom-up construction. So building a synthetic cell from non-living parts using Mycoplasma cell-free system, as Tyler also presented to us earlier. So each one of these routes offer complementary ways to test feasibility and actually bringing our phytosyn to life. So we hope we can manage this, but last but not least, we still have to manage to put our little feijoada inside protoplasm. So this is another step that we have to overcome. And to integrate phytosyn directly with plants, we are considering protoplasm fusions, different protoplasm fusions methodologies. So protoplasts can be isolated from any current plant synthetic biology models, from Arabidopsis to crops like maize and tomato. So we can have a very, different plants that we could try and use. Of course, this also will be matched with the phytoplasma species that we are going to choose for this endeavor, for this test. But once we decided which protoplast we will use, so we'll perform fusion we are going to we will test 3 different methodologies to perform the protoplast fusion with the phytosyn. So you can be promoted chemically using PAG, electrically using electrofusion or even through microfluidics, where single protoplasts are aligned and fused in controlled channels. So these three approaches, I believe that will allow us to test the feasibility of molecule-plant fusion, and of course, explore the phytosyn as a synthetic endosymbiont. And well, as a conclusion of this adventure that we're just starting, so this is a project under construction, but actually it opens exciting perspectives, which is advance our knowledge of plant microbe interactions. These I didn't mention, but phytoplasmas actually are harm to the plants, so it can cause severe loss, economical losses in agriculture. So this will be very interesting. These tools also help us to understand a little bit the biology behind phytoplasma pathology. Of course, pioneer synthetic biology platform for agriculture and contribute to climate smart strategies and sustainable farming. So our next steps, and of course, in conclusions, if you actually perform these, we're going to be building the first minimal phytoplasma cells that could represent a new frontier in plant biotechnology. So we aim to shift phytoplasmas from threats to opportunities, and ultimately build tools for the future of crop improvement. And with this, I thank you all for your attention. And I would like to thank you mostly for Vincent and John Glass for the partnership. And we hope that we can have other folks to join us in this endeavor. Thank you very much.
Questions for Danny?
It seems like, one of the very severe problems working with the phytoplasmas is that you can't culture them. And I was wondering, if there had been progress that I'm unaware of in that space, or if, what is happening there.
Yeah, well, there are some papers published, but very, very written up, very good yields. But one of the things that we are planning is actually mixing and matching maybe some other metabolic pathways, maybe from SYN3B, from mycoplasmas as well, to see if we can make this work. And there are also another group in France that is actually performing some metabolic studies, trying to overcome this issue. But I think this is going to be the most challenging part that we're going to have to overcome.
One of the things that I've been thinking about, is that, the phloem, context is unique in that it has a high pressure environment. It has about a 25 bar pressure in the phloem, and I don't know whether people have tried culturing phytoplasma at higher pressures, but that might be something to try.
Oh, thank you, Tom. Actually, this is a good thing to check it out. Thank you very much.
Just a few comments here, because maybe, again, potential another collaboration. So we are doing some of that work, but it is with, again, with the E. coli plasma. So we start exploring the fusion with plant cells as we speak, and we start cloning small pieces of phytoplasma in E. coli plasma. So, kind of the same route, because, kind of for the same reason, could we park it and then, make this hybrid E. coli plasma phytoplasma cell. So, there are potentially some overlaps that maybe we could discuss, but
for sure.
But that is very kind of interesting, obviously the genetic code and everything, compatibility. We're starting on with few genes, but E. coli plasma, because our goal was turning E. coli plasma into phytoplasma E. coli plasma hybrid with all the tools. So maybe that is something for us to potentially explore. Yeah.
Oh, great, Bogomil. I'd love to. Thank you very much. It's going to be great. Like, it's something that you think, oh my, this is going to be possible. Should I do that? But someone once told me, actually was Liji once in the lab. She told me, well, if you don't try, how are we gonna know if it's possible or not? So I think we have to go for it and see if we can accomplish. So, and of course, all the help is very, very welcome. Thank you.
Yeah. Yeah, and the cell fusion, we're also exploring them with diatom protoplasts. And so far, E. coli plasma fuses really well with yeast, obviously other E. coli plasma, but we are exploring plant and diatom cells right now. And, if that works, maybe we could put some efforts to try hybrids, mycoplasma, phytoplasma, or E. coli plasma, phytoplasma
Yes, for
hybrid.
sure, for sure.
Yeah.
Thank you.
Okay, I'm really excited that there are more people.
Yes, yes. Well,
Yeah.
let's set a meeting.
Yeah, thank you.
Thank you.
Daniela, it still seems to me that while even if you develop great methods for phytoplasma genetic manipulation, and to me this seems like probably a place you're going to have to spend some time, is thinking about making a cell-free phloem for phytoplasma replication. Or,
Yes.
and I don't know if it would be necessary to think about putting that under high pressure, but the technology for that certainly exists. But okay, so let's assume that you become, a brilliant engineer of phytoplasma genomes and biology. I'm still thinking about your nitrogen fixation issue. And in nitrogen fixing bacteria, do they have an organelle for nitrogen fixation that allows them to sequester the nitrogenase from oxygen?
It has been shown in algae, John.
And so
there is
is there knowledge to think about trying to engineer such an organelle in phytoplasma? Because if you put all of the nitrogenase system in a phytoplasma cytoplasm, I'm sure that it's going to be oxygen poison. I'm confident, but not absolutely sure, but confident.
Well, Danny, do you mind if I jump in here, Danny, real quick?
Of course, Vincent.
Hello, everyone.
Thank you.
Yeah, John, regarding your question for example, for
Vince, yes.
Oh, hey. So for cyanobacteria, right, a lot of the filamentous cyano will actually undergo differentiation and form these anoxic cell types, right, these heterocysts. And I think that the interesting thing with phytoplasm actually is that the phloem is actually fairly hypoxic, right? And then at the same time that they actually, when they get transmitted into the insect gut and live in the hemolymph for a while, it's pretty much similar to the phloem and as well anoxic. So at least we don't know yet if it's poisoned by oxygen. We do have an idea that it's living in a very low oxygen state, which is why I think we're a little more hopeful that nitrogenase could be potentially useful here. They've shown nitrogenase works in aerobic cyanobacteria like Seneca cystis, so there's some hope, but they don't get the efficiency that you would from, say like it's in its true environment.
Or the irony would be, that the nitrogenase system only works in the insects. So you have nitrogen fixing insects that suck on the plants, which is maybe not going to be a commercial success.
Yeah.
But
that's a good point.
Yes, probably.